The first person I'd like to ask to come up is Prof Phil Bremer, who is a professor Distinguished Professor in Food Sciences at the University of Otago and Chief Scientist at the New Zealand Food Safety and Science Research Centre. So he looks at food safety research across a huge number of sectors, and I will ask him to come and talk all about what they discovered.
Thank you. I'd like to start off by thanking Dan and Sally and the other organisers for organising the event today and for inviting me to talk to you. I'm delighted to be here to share some of our perspectives. Some slides up there, please. I want to start off just briefly talking about what the New Zealand Food Safety Science and Research Centre is. It was established in 2016 after the WPC 80 incident with Frontiera. And it's a partnership between industry, regulators and 8 research organisations in New Zealand. It's a virtual centre. We have a very small management team, and our goal is to talk to industry, understand their problems, and then go out and find the best science teams to actually address that. So we have good links across all these sort of the researchers working in the food safety space in New Zealand. And our goal is very simple. We want to increase and maintain confidence in New Zealand's food safety systems, maintain and enhance our exports. Now I want to start off with a disclaimer. As the cyclone was carrying out up here, I was in my office in Dunedin, and my only real involvement with the cyclone was via phone calls or Zoom calls. However, my fellow co-authors on this presentation have told me that the following days and weeks were quite chaotic. I have a number of people I'm presenting this research on behalf of. Because we're a centre, I've collected the views of a large number of our members. So we had some scientists on the ground from Plant and Food Research, now BSI, we had ESR researchers involved, and I've also picked up the views of the people from New Zealand Food Safety, MPI, that were involved in the response. Also at the start, I'd like to acknowledge the supporters and funders of this research: New Zealand Apples and Pears, Zespri, Vegetable New Zealand, United Fresh, the Food Safety Science and Research Centre, the many Hawke's Bay orchardists who allowed us to go on their land, and the government who funded some of this research through emergency funding. As you know, the cyclone presented many challenges. We've heard about the widespread infrastructure challenges that impacted upon transport and communication. And you're all very much aware of the emotional, physical and economic concerns and challenges that this caused to producers and their operations. What you may be less familiar with or given less thought to is that the challenges this incident imposed upon the regulators. They've told me that they found this a very stressful time. They needed to simultaneously address multiple concerns across multiple stakeholders. They weren't only dealing with the horticulture industry, there was the red meat sectors, there were consumers, there were butchers, there were grocery stores, and everybody had their own concerns. And the MPI people were doing this on top of their normal expectations. One of the other challenges about the cyclone and its damages were its very large scale and the inability to make generalisations, as the risk profile for each site and each crop differed. And there was also a lot of disagreement around what to do and what to test for. Are we worried about the sediment that's coming off the hillsides, and what can this contain? Is it going to be diluted? Or are we worried about the water that's flowing through the sewage treatment plant or the fertiliser plant or the chemical store and the impacts that this will have? We really had no capacity for testing. We couldn't get the test kits into this area, let alone get the tests or the samples out to be tested. And amongst all this, the regulators and the scientists had to deal with misinformation and emotive communication, and people were talking about toxic silk and saying things such as E. coli numbers are through the roof. We've heard a little bit from Dirk and others about the challenges for the researchers, and some of the challenges that we face, that there was limited previous research or publications or guidelines on where to base our recommendations. And this came as somewhat as a surprise, okay, there's a flood, what do we do? What do we know about it? And in reality, we knew very little as a research community, either here or internationally. We didn't really know what the background levels were, background microbial levels in our soils were. Therefore, if there was high numbers of E. coli, we didn't really know if this was a big deal or not. How do you know if there's an increase if you don't know what the background levels are? There's also pressure to produce scientific advice rapidly, without the sort of things that academics and researchers like to do, send it out to peer review, have a think about it. People wanted results the next day. The working conditions were difficult, we've heard about that already. We had limited access to sampling sites and sample collection equipment. And the people who are on the ground here told me they found it quite challenging actually working with the orchardists and the growers who were under such enormous stress for themselves. So what was our response? The initial response, owing to a lot of the researchers and research organisations donating time and money, we managed to, with government funding, get within 5 days some best practice recommendations to be issued, and then we started some desk-based and on-the-ground assessments. To ensure safety, the decision was quickly made to condemn all ready-to-eat produce that had been in direct contact with floodwater. This was a very sensible decision, and it's in line with any sort of recommendations that other regulatory agencies around the world have. Now, one of the things that may surprise you was that well-structured food safety testing programs only started 1 month after the cyclone ended. And this was for a number of reasons. One was obviously priority was given to people's well-being, but in reality there's also really a lack of capability, and there was challenging conditions, as I already mentioned. We just couldn't start the test. So there's a whole month of data. I was looking at the NIWA data and all that online system and water quality data. We'd love data like that. We just couldn't get it. So what were we concerned about? Well, we were concerned that the floodwaters may have contained pathogens, so bacteria and viruses. It may have contained chemical contaminants, and it may have contained, or we knew it contained, physical hazards. And these hazards could have come from a wide variety of sources, from sewage, from animal waste, from dead animals, decaying plants, displaced agrochemicals, displaced sediments and the heavy metals they may contain, spilled petroleum products, and then the physical hazards were things such as building debris, damaged equipment, and wood chips. And the questions of immediate concern for the horticultural sector included, and just firstly, was produce suspended above floodwaters on trees and vines safe and suitable to harvest and sell? We wanted to understand the risk that these products pose to the consumers and to the markets in terms of actual food safety, but also reputational risk. Want to understand if there are any steps that the horticulture industry could take to help to ensure the safety of such produce. And then the sort of second lot of questions were, how do we know when it's safe to replant our leafy greens that have been in water that has been submerged? And a question that we didn't actually even get around to addressing, I don't think we've addressed so far, was could pathogens from the flooded soils be translocated through the roots into the produce? So I'm going to start off talking to those first few questions around apples and kiwifruit, and then I'll talk about replanting flooded soils later. So the first thing to try to understand was the produce that came in contact with the floodwaters, were above the floodwaters, was this actually safe to sell? And if it wasn't safe, what sort of pathogen numbers were there? So our scientists went out, this is plant and food research scientists, they went out and sampled 12 orchards across the region, obtained apples that were actually below the waterline from 30 centimetres and then at various levels above the waterline. And as you can see from the image there, it was usually quite easy to see where the waterline was. And then they sampled weekly for 4 weeks, starting 4 weeks after the event. So 1 month passed, collected samples, and then went out every week after that. And what did we find? We looked for typical sort of pathogens that are of foodborne concern, looked for E. coli, and strangely enough, we thought it was strange, there was no clear pattern of prevalence whether the apples were actually below or slightly or well above the flood line. We didn't detect Salmonella or Campylobacter, other important pathogens, and Listeria monocytogenes was only detected in 1% of samples, and that's probably not too higher than what we'd normally expect. Importantly, with Listeria monocytogenes, which as you're probably aware is an important pathogen, numbers did not exceed the sort of the guidelines that we normally have of 100 bacteria per gram of produce. And we know that if we do have a few Listeria on apples or kiwifruit, we know that these products the bacteria don't grow on these produce, therefore the low numbers, we believe, don't pose significant health risk. We also went out and tested kiwifruit for the presence of pathogen, went to 10 orchards, tested 10 bays in each. We sampled fruit that had no contact with floodwater, fruit that had possible contact with floodwater, and then fruit that we knew had direct contact with floodwaters. Of 313 samples that we tested, only one sample reached the Food Safety Australia New Zealand requirement for rejection in terms of E. coli. We did find E. coli on it, and we knew that generally the E. coli numbers were greater on the green, the Haywood kiwifruit, than on the gold, and it's sort of put down to the fact that the green is hairier, as you all know. Whether that's a true fact or not, I don't know, but that's the general accepted reason for why we get slightly higher numbers of pathogens on green compared to gold. Went and tested 263 samples of fruit for Listeria monocytogenes. None exceeded the limits for its presence. And however, in general, we did find that contact with floodwater slightly increased the prevalence of E. coli and Listeria monocytogenes on kiwifruit. But remember, all kiwifruit that had contacted floodwaters were actually condemned and not sold anyway. We did not find Salmonella, Campylobacter, or sugar toxin E. coli on the kiwifruit, regardless of whether they'd been in the floodwater or not. So the question was, well, why was so little contamination detected on flood-affected fruit? We knew that this fruit had been in floodwaters. Why didn't we find these pathogens that we're so concerned about? And really, as I think you're probably all aware now, this flood event was enormous and the dilution effect was also enormous. So effectively, if it picked up any bacteria through contact with fecal material or the land, they were just diluted out. That's one reason. The other reason is that we didn't start looking for it until 33 days after the event. So maybe it had been washed off by rain or being killed by UV. But we do know that we can normally find E. coli and Listeria on these types of produce, so we do know they can survive on there if they get there. New Zealand apples and pears use this as an opportunity to actually test their mitigation systems. Normally it's quite difficult to actually find naturally contaminated fruit at high levels, but we had this opportunity to actually do that. So they picked apples that had been under the floodwaters and hence had higher levels of pathogens than normal, and they went and tested are there processes that we can implement to actually remove those pathogens or reduce the risk that this hazard poses. And it was found that a high pressure wash, just their normal standard high pressure wash, significantly reduced E. coli numbers and eliminated Listeria monocytogenes from the few samples where it was found. And then they went a step further and tried to understand if we add sanitizer to our wash water, do we get a better effect? So they were trying to learn from this flood event. And on the graph, the top graph, for example, the orange bars right at the far edge here, this is the control after 34 minutes washing time. It's going to come through as clear as I would have liked. And effectively, in the absence of the sanitizer, there was relatively little reduction in E. coli numbers, but in the presence of the sanitizers, as shown by the pinky sort of lines, E. coli numbers decreased dramatically. So we knew that these two commonly used sanitisers, harvicide and prussic acid help to reduce pathogen numbers on the fruit. We also did some work around what washing temperatures were better, and for example, washing at 15 degrees was better than washing at 7 degrees. So the conclusions around the impact of the flood on food safety were the risks that the fruit posed. Overall, the food safety impact, whether it be pathogens or chemical contaminants, on the fruit was low or not measurable. Dilution and time after harvest may have been a significant factor in this. So while I say that for this particular flood event, the risk doesn't seem to have increased dramatically, it's important to appreciate that each flood event is likely to pose its own unique challenges. And good harvest practices are important regardless of the flood event and the risk that it may pose. So the recommendations are no harvesting of fruit lower than 30 centimetres above the flood level, avoid or minimise getting mud or silt on the fruit during harvesting, bin cleaning is vital, and we've heard this already today, I know, and if appropriate and if your product allows it, soaking or washing or using high pressure washing, especially in the presence of sanitisers, will help to reduce pathogen numbers and further reduce the risk. Now, in terms of research on flooded soils, much to our surprise, when we went out to try to understand what best practice is, we found that there was really limited information internationally around what's best practice, how to remediate, when to replant flood-affected soils. We carried out two sort of research projects. The first was we commissioned ESR to carry out a lit review to determine when flood-affected soils could be affected. And the sort of questions we asked them to address was the impact of flooding on fecal indicators and pathogens in horticultural soils, the best approach to assess microbial safety of horticultural soils, and if we can use other markers, we can't detect the bacteria themselves, can we look for fecal material, markers of fecal material, to help us understand when that soil may be safe. And we also carried out some research. ESR tested 20 soil or silt samples, which are kindly supplied by Landwise. They looked for common pathogens, E. coli, Salmonella, Campylobacter, sugar toxin E. coli, Yersinia, Listeria monocytogenes, and some of these microbial source tracking markers. This report has been published. It's available on the New Zealand Food Safety Science and Research Centre website, and I'm just going to talk about a few highlights from it. So what do we find out about what's normal in horticultural soil? And this is based on relatively little data. E. coli is usually not detected, and if it's detected, it's normally present in low numbers, but sometimes it can be present in very high numbers, so greater than 1,000 colony-forming units per gram of soil. So the detection of E. coli doesn't necessarily mean something is wrong, or the presence of E. coli high numbers doesn't even necessarily mean something is wrong. You need to understand what's the normal situation of E. coli. So E. coli can be an indicator of fecal contamination, but E. coli can also be a normal component of your soil. Listeria monocytogenes is normally detected at about 10% of soils. So if we go out and look for that during a flood event, what is that really telling us? Information also tells us that irrigation and rainfall tends to increase the likelihood of E. coli and Listeria monocytogenes being detected. And this is sort of our traditional wisdom. E. coli's in the gut of cows defecate on the paddocks. With rainfall, the fecal material gets picked up, so you get high levels of E. coli. We sort of know that, we understand that from a food safety point of view, but we don't really understand that from what's normally in the soils. Some of the more pathogens of concern from a health point of view, Salmonella, sugar toxin E. coli. This is the E. coli that contains the toxin genes that make you sick. So there's the E. coli, that's a normal inhabitant of our gut, harmless, and then there's the sugar toxin E. coli that's picked up the virulence factors that produce a toxin. Sugar toxin E. coli should not be present in soils, and if it is there, then that's probably more of a concern than generic E. coli. So what happens to soils after the flood event? Well, not surprisingly, initially there's elevated numbers of pathogens, but then these numbers decrease over the subsequent days or week. So this is international data. A lot of the research has been carried out in the US. We know that higher soil moisture encourages persistence, and pathogenic microorganisms are more likely to be present in soils if they're also present in nearby surface water. And of course that makes complete sense. If the pathogens are in the flood water, then they're likely to be in the flood-affected soils. There has been work done looking at survival of pathogens in soils, but it's mainly been done in the context of actually adding manure to soils in order to facilitate the health of the soil. And when researchers have added pathogens, manure containing pathogens to soils, they've seen an initial rapid decrease in pathogen numbers, followed by a longer period of slower reduction in numbers. So if we can sort of correlate or use that manure-based data to flood-based data, quite a different inoculation procedure, but pathogens are in the soil, what does that actually tell us? What tells us that bacteria, pathogens, can survive up to 2 months in soils, maybe 3 months. We know that temperature is a major driver. They die faster as the temperature increases, and the higher moisture content prolongs survival. Soil type also plays a role with poorer survival in coarser sandy soils, which no doubt dry out faster and heat up faster. The other thing that was somewhat of a surprise when we were trying to understand best practice was to learn that there's no real standard methods to assess the microbial safety of horticultural soil or even flood-affected soil. There's sort of two standard ways. One, you can go and get a little trowel and dig a bit out, and this samples both the surface and the subsurface, but it's quite time-consuming. The other sort of option is to drag a gauze around or put on some shoes which have gauze on the bottom and stomp around and collect it up that way, but that only really picks up the pathogens on the surface. Is it important to till to break up flood-compacted soils? Tilling is obviously important to help to restore the soil to a productive state. However, it increases soil aeration, and you incorporate these flood-deposited microorganisms actually into the soils, and this will probably enhance their survival once they're in the soils rather than on the surface and exposed to UV. And it may even help some pathogens to grow and increase in numbers. However, based on the limited data and the importance of actually remediating the soil, we still recommended that soil tilling is important as soon as you can get onto the land. The benefits of getting the soil remediated outweigh the risk that this pathogens will survive for longer. Another question we had was, can we actually look at other markers, some fecal indicators, genetic markers, markers for cows or pigs or goats, for example, to understand are we truly getting fecal material into their soil. We found that for E. coli, a concentration of greater than 1,000 mpm or numbers per gram suggests the presence of fecal material. However, this was not a reliable sole indicator of the safety of soils for replanting. As I already said, we don't know what the initial numbers are, so it's very hard to know what increased numbers mean. If we combine this with some microbial source tracking, so E. coli associated with cells from cows, may be an indicator that the paddocks have been contaminated, and may be a better indicator of fecal contamination and whether the soil is safe or not. Listeria monocytogenes is more common in flood-affected soils, but we're really unsure how its presence correlates with fecal contamination. E. coli and Listeria are microorganisms that live in different environmental niches, and just because you find one doesn't mean the other will be there and likely to pose a problem. So testing for Listeria monocytogenes, which is a concern in the fresh cut industry, is not recommended to inform replanting decisions. However, if you do find it, it does mean that maybe you have to take better care if you're processing something like the leafy greens, but it shouldn't inform your replanting decisions. So, conclusions, around replanting flood-affected soils, a 60-day period between floodwater receding, the land drying, and you'll be able to get on it, and replanting is consistent with all available evidence on pathogen survival and most other advice. I will, however, say that there's not a lot of science, not as much science as I would like behind this, but it's pretty consistent advice that we see around the world. If you need to plant or want to plant faster than 60 days, then you'll need to go in and prove to yourself that that soil is safe, and that will require probably some background knowledge, what is normally there, and some tests to actually look at pathogen levels and the presence of these microbial markers. I've already talked about the importance of prior knowledge of what is normal, and this will help in hazard identification and risk assessment and help to inform your decision making. Just want to finish off talking about getting prepared, and this is obviously a very important aspect. To better prepare for future adverse weather events, horticultural industry stakeholders must, and I say must, improve their understanding of the current risks, so what is normal in their soils, understand the sources of potential contamination, and look at what risk mitigation steps they can take. And apples is a classic example where you can incorporate a mitigation step in terms such as washing. You do understand, practice scenarios where adverse weather events such as flooding will do to your products, and understand how does it actually change the hazards that may be present and therefore the risks to your product. Will it introduce new hazards that are not normally there, or will it simply increase the number of current hazards or the concentration of the current hazards? It's important to invest in people. Everyone we've spoken to has stressed the importance of developing a good food safety culture, and I've been told by many growers and people on the ground that they cannot overstate the vital role that employees focused on doing the right thing played in ensuring the safety of the produce that they were producing. Need to also invest in developing scientific knowledge and capability. You may say, well, of course you would say that, he's a scientist, but in reality, there are so many gaps in our current knowledge that if the same event happened tomorrow, we'd probably be no better place to give you advice than we were 2 years ago. We haven't really moved forward. Preparedness is key. Having the knowledge and technical capability to roll out instantly will mean you can respond quickly. You need to have a plan in place. You need to practice your scenarios so you can act. There's a quote I heard yesterday, and it was that in the presence of a crisis, we all like to think that we will rise to our expectations. However, in reality, we fall to the level of our training. So you might think you'll react to this, but you'll resort to what did you train, what do you know, and obviously if your training is not good, then maybe your response won't be so good either. So preparation is the key. This includes knowing what to test and what to test for. Do you test the produce? Do you test the soil? Do you test the water? Can you guess how many flood samples we tested for the presence of bacterial pathogens? Who thinks it's more than 100? More than 1,000? Less than 50? Who thinks we forgot to take any samples at all? As far as I know, there are no floodwater samples taken. Now, maybe that makes sense, or maybe it doesn't, but we have no idea what was in that floodwater. People were busy, they're doing other things. The test kits to do sampling come down on a truck routinely from Auckland. Could they get in here? No. The samples that were being taken get sent up to Auckland to get tested. Could they get out? No. Could the scientists get in? No. We didn't have the capability to test even if we wanted to, but people were too busy to think. It wasn't until later. So you need to understand what to test and what to test for. So what produce do you test? What samples do you test? Do you look for microorganisms? Do you look for chemicals? Do you try to find E. coli? Is it presence and absence, or is it numbers? If you get numbers, what does it actually mean? How how to test, what are the sampling protocols? Do you test fruit above the floodline, below the floodline? If above, how far above? And do you know actually how to test? If your normal samplers can't get into the field, could you go out, pick up some sampling kits, do the testing yourself? Do you have sampling kits in the cupboard that you can use? You know, how do you get more prepared to actually be able to answer some of the important food safety questions that enable you to continue to put your produce onto the market? The researchers weren't available, couldn't get into the area. The testing labs in New Zealand are always pretty much acting at full capacity, so there's no way they can handle an extra 1,000 samples or whatever. So even if we could test, we probably couldn't find someone to actually carry out the results for us. And finally, there's a cost associated with testing, and you need to understand who will pay, how this data going to be reported. Right, finally, some take-home messages. Preparation is obviously key, and as the Cyclone Gabrielle response was coming to an end and we're having our wrap-up meetings, virtually everyone I spoke to in the food safety space said we will do better, we will get prepared, and the people who have asked in this space haven't really done much. We haven't really progressed anywhere near as much as we'd like to in this space. However, regardless, preparation is still the key, and it's not too late. You can start getting prepared tomorrow if you're not already prepared. So we need to have the knowledge and technical capability to roll out instantly. If you can do this, you can respond. You can get ahead of the game. Data is fundamental to any crisis response, and in a crisis, would you know who to turn to, what needs to be assessed, how to assess it, the standards that need to be met. Do you have industry best practice? Are they agreed upon? How do you meet those? And we've heard this already today, communication. Who actually speaks for your industry? Who's the one voice of reason or the one voice of truth that the media, your customers, overseas markets can actually turn to understand the response? And if you can answer all these questions positively and on top of it, then you're doing well and you're able to respond more effectively to a crisis than others. And just going down to this last point, growers, horticulturists, farmers, you're all in the process of producing food. And if it's not safe, it's not food. Food safety is paramount. And our strong message would be that if there's any doubt that the product is safe, you can't sell it, you have to condemn it. Thank you very much.